on to lipids, and lipids are basically primarily composed of hydrogen and carbon, not a lot of oxygen. And because of that structural property, because of that structural difference from carbohydrates and even proteins, as we'll see, because it doesn't have as much oxygen, it basically creates this uneven sharing of electrons around this molecule. And so what that creates is polarity with lipids. So lipids are basically non-polar, they're hydrophobic, they don't play nicely with water. And I'm sure we've all observed this. If you put a drop of oil, it's going to completely separate from water. And that's because it's non-polar. It's got a nice, even, stable charge around that molecule. Whereas other molecules that have a lot of oxygen, they have an uneven sharing of electrons. They are polar. They have an uneven separation of the charge across that molecule. And so polar molecules like carbohydrates and like proteins are going to play nicely with water, hence the word polar. Whereas non-polar molecules like lipids or fats do not interact nicely with water because they don't have any sharing of electrons around that molecule because of the presence, or rather the lack of oxygen.
Go
ahead.
Sorry, okay. I was actually going to say, what's the difference between lipids and fats?
Lipids are fats. Those are synonymous terms. They're different. Same thing. Okay. So we've got 5 classes or 5 types of lipids: triglycerides, ketones, phospholipids, eicosanoids, and then steroids. So we'll go through those individually, but they're all pretty much, they behave the way that all lipids do, in that they're nonpolar, hydrophobic, do not interact or play nicely with water or in aqueous environments. Let's start out with triglycerides. So triglycerides are another important storage unit for energy. So when we eat a meal that has fats in it, they're going to be broken down to the smallest components, which are triglycerides. And then within our body, we can actually store these in different parts of our body, usually in adipose cells, for energy to be used at a later time. Triglycerides are made of glycerol, which is a glycerol backbone here. And then it's got 3 fatty acid tails attached. So we've got this glycerol backbone, and then these are 3 fatty acid tails that are attached. And this is the basic structure of a triglyceride. The glycerol is basically a 3-carbon alcohol, which is not super important. Then the fatty acid tails can vary quite a bit, and it's going to give some different properties to the different types of triglycerides. So it's basically a long carbon acid chain that's attached, respectively, and it can be different in terms of the types of bonds that are found along that carbon acid chain. We can have a saturated fatty acid, which simply means that as many carbons are bound as is possible. It's completely saturated. There can be no other, there's no other opportunity for binding on any of the carbons. They are fully or maximally saturated. Then we can have an unsaturated fatty acid. An unsaturated fatty acid does have a double bond. So we've got a double bond here. These are 2 examples of unsaturated fatty acids. And what that simply means is that they do have opportunity for additional binding where we have these double bonds. So we have saturated versus unsaturated fatty acids. And among unsaturated fatty acids, we also have other smaller differences structurally. So if we have the hydrogens bound on the same face of that double bond, we can call it a cis fatty acid. An example is going to be oleic acid. And if we've got the hydrogens bound on opposite ends of that double bond, that's going to be a trans fatty acid. And an example is going to be alideic acid. And so essentially, we see very small structural differences between these types of triglycerides: unsaturated versus saturated, cis versus trans. But these small differences lend to very big functional properties that we see with these different types of triglycerides. So for example, we know that traditionally unsaturated fatty acids and more so trans fatty acids are more hazardous to our health. They can be more of a problem in terms of blocking arteries and creating plaque along our vessels. Excuse me. And when we look at the basis for the difference in these two structures, it's as simple as the difference in where that hydrogen is bound. And so that very small structural property or small structural difference actually brings about a very large functional consequence or functional difference with these different types of fatty acids. The second type of lipids are ketones. And ketones are basically byproducts of free fatty acid that are released into the blood. They become important when we speak about diabetics or persons who are on a low carbohydrate diet, often referred to more commonly in the media and so on as a ketosis or a ketotic diet. And so this diet is very low in carbohydrates, and because it's low in carbohydrates, we don't have our typical source of glucose for energy, and instead we have to tap into our fats and adipose tissue to supply our cells with energy. So we basically hydrolyze our triglycerides. Remember, I talk about triglycerides as being the main storage form of our fats in our adipose cells and adipose tissue. And so because there's no carbohydrates or glucose circulating, for example, if you're diabetic or on a low carbohydrate diet, we now tap into our fats to release those fatty acids for energy. And so the byproduct of releasing those fatty acids are basically our ketones, which are produced from the liver. Now, this can be detrimental or hazardous, especially in diabetics. And I'll kind of explain why diabetics have low carbohydrate or low glucose levels. In diabetes, whether it's type 1 or type 2, there's an inability for our cells to uptake glucose because of the presence of the hormone insulin. So insulin is the hormone that sends the signal to our cells to uptake this glucose from the bloodstream so that it can be used to create energy within our cells. And so someone who has diabetes is either not going to have as much insulin in the case of diabetes type 1, or they're going to be insensitive to that insulin in the case of diabetes type 2. And so on either end of that spectrum, the end product is that they cannot uptake their glucose for usage inside cells. And so they're basically going to tap into other storage and other storage forms to create energy. And for diabetics, this buildup of ketones, right? This buildup can cause ketosis, which is basically an excess state of ketones in our blood, and that can lead to ketoacidosis. Ketones are quite acidic, and they can raise the acidity of our blood or lower the pH of blood, and that can obviously be quite detrimental to neurons, to muscle, and so on. I'm seeing an interesting contribution here in the chat. So someone did a research project, and it had to do with the biochemistry of ketones in the diet and it being researched as a treatment for epilepsy. So that's a very interesting connection there. There has been a lot of research done on ketotic diets or diets that are low in carbohydrates and allow this liberation of fatty acids and the production of ketones as being quite therapeutic for a lot of different diseases. For epilepsy as one, other congenital disorders where there's an inability to break down other forms of glucose, they have quite a lot of success on ketotic diets. And so ketones and the presence of a lot of the ketone bodies and a low carbohydrate diet, which produces more of these ketones, can be therapeutic in some instances. Okay, so it's a very excellent connection that someone pointed up there. And then the last bit here really is the way that this excess ketones present is through this acetone breath. So because ketones are quite acidic, they give this very harsh acidity or this harsh kind of a metallic taste or metallic smell rather to the breath of people who have excess ketones in their blood. At the bottom here, we're looking at the structure of ketones. And so it's basically this carbon double bound to an oxygen and then two functional groups attached. Now these R's can represent any number of different functional groups, and that can give us different types of ketones. And so one example that we see here to the right is acetoacetic acid, which has a hydroxyl group as one of the functional groups here, and then a methyl group as another functional group. Right, so we can have a whole host of different, types of functional groups attached to these carbons that will give us different ketones altogether. Okay. The third class of lipids are phospholipids, and phospholipids are probably the most important lipids that we will encounter. These have a property of being amphipathic. And what amphipathic means is that one part of the molecule is polar. In other words, it likes to interact with water, the heads of these molecules, and the other part of the molecule is nonpolar. In other words, it is behaving like a traditional lipid in that it doesn't interact well in aqueous environments. And so a molecule that has this makeup, that has this structure where one part plays nicely with water, one part doesn't play nicely with water, is the perfect structure for our cell membranes. It's the perfect structure to create those compartments that we've been talking about in our previous packet, right? The fact that we can have fluid inside our cells, fluid outside our cells, but have this barrier, this cell membrane that kind of compartmentalizes these two aqueous environments is really brought about by the structure and the amphipathic nature of our phospholipids. Okay, and so when we look at the structure of our cell membrane here on this slide, we can see that the polar heads will face the aqueous environment. This may be the inside of the cell, whereas the nonpolar parts of this phospholipid, the nonpolar tails, are going to face each other. And so that's going to create a barrier where we've got the heads facing the aqueous environments because the heads are polar and they can interact with water, but then the tails are facing each other, and that is creating the barrier itself. Another example of where we see phospholipids are micelles. Now we'll talk about micelles probably way later on in semester 2 when we talk about lymphatics and how fats are absorbed. But when we think about fats moving throughout our system, because they are nonpolar, because they're hydrophobic and they don't play nicely with water, generally, things like our triglycerides and our steroids, which we'll talk about coming up, we must be able to package these fats in a structure that does play nicely with water. Our body's made up of so much water, and in order for these fats to move throughout our system, to move throughout our vasculature, to be absorbed and secreted, they have to be packaged in what's called a micelle. So the fat would be stored in the internal part of this micelle, where it'll be in contact with the hydrophobic or non-polar tails of these phospholipids, whereas the outside of the micelle has the polar heads, which are moving along quite nicely with the aqueous environment. So we can see how a structure like a micelle would be pertinent to moving fats around in our aqueous physiologic environment. Another example of phospholipids is surfactant. And surfactant actually prevents the collapse of our lungs. It's secreted during the fetal period and right before birth. And what it does is it helps to decrease the surface tension that's otherwise seen within the alveoli of our lungs and keep them nice and patent and prevent them from collapsing. And so surfactant is going to be really important to maturing the lungs and preparing that fetus for birth so that they can take their first breath as soon as they come through the birth canal. Okay. Otherwise, during the fetal development, during the fetal developmental period, the lungs are collapsed for the most part. And then right before birth, we've got this release of the surfactant, which opens up the lungs, keep them nice and patent, and prepares that fetus for birth.